WorldCommunityGrid.org has been harvesting spare processing time from computers around the world, using that computing power to accelerate research that helps humanity and the world. We thank our members for contributing over 500,000 years of computer time to the important projects on WorldCommunityGrid.org. About 6 years ago, the second research project to run on World Community Grid was Fight AIDS at Home. This project is searching for drug candidates that could inhibit the HIV virus that causes AIDS. Please let me introduce Dr. Alex Perriman at the Scripps Institute in La Jolla, California. He will tell you about the science and recent progress in the Fight AIDS at Home project on WorldCommunityGrid.org.
Hi, Victor. Thank you much for the introduction. I'm Dr. Alex Perriman, that's my photo there, and I'm a member of Professor Art Olson's lab at the Scripps Research Institute in La Jolla, California. Now, on the next slide, I'm going to tell you about the significance, why we're actually working on HIV. So far, there are over 34 million people who have an HIV infection, about 3 million people get infected each year, and about 2 million people die from AIDS each year. In total, over 30 million people have died of HIV-related causes. On the next slide, I'll tell you about the itinerary for today's webinar. So first, I'll tell you about how World Community Grid helps us all. Then I'll present some progress on some virtual screens against HIV protease, where we discovered 2 active compounds in experiment 28. Then I'll tell you about an extension of this experiment. I'll move on to some new virtual screens against HIV integrase that are searching for allosteric inhibitors. And then I'll tell you about what happens after these calculations if we have time during the webinar. On the next slide, I'll tell you about how World Community Grid helps us all. It provides free computational resources from nearly 2 million different computers from throughout the world for projects that benefit humanity. Now, these donated CPU cycles help us advance research that seeks faster ways to cure diseases, find renewable energy materials, create clean water techniques, or develop healthier food staples. Now, in about 6 years, over 145,000 CPU years of calculations have been donated to our Fight AIDS at Home project. Now, on the next slide, I'll tell you about what kind of calculations we actually do in this Fight AIDS at Home project. So on Fight AIDS at Home, we do what's called docking. It's a computer calculation that tries to find master keys to unlock the control panels that can stop each particular piece of the viral machinery. But to make things more complicated, the keys and the locks are both flexible, they change shapes, and they have some charge parts that must match up well too. To make it even more complicated, there are often multiple different keyholes per lock, and only 1 or 2 of those keyholes might actually work and give you the desired ability to stop the virus. And another layer of complexity is that there are over 10 to the 60th power different potential keys in this chemical space, or all the different types of compounds that could exist. So that's a 1 with 60 zeros after it. Now, we can obviously only calculate how the anti-HIV potential of a small percentage of these 10 to the 60th power keys, just a few million compounds. So we try to pick the compounds that are most similar to compounds that have already become approved drugs. On the next slide, I'll tell you about why we're focusing on HIV protease. So HIV protease is a key viral enzyme that cuts the viral multi-protein chain into its separate pieces, which then allows those pieces to fold up into their normal 3-dimensional structures, which allows the viral particles to form, mature, and infect other cells. Now, if you block HIV protease strongly enough, then that causes that infected cell to produce immature, non-infectious particles that are able to be removed from the infected patient. The most important thing is that the introduction of protease drugs to the anti-AIDS cocktail significantly reduced the death rate associated with HIV infection. Now, on the next slide, I'll present some clinical data that actually proves that inhibiting HIV protease does help treat patients who have AIDS. So this is a slide from Alex Vlodauer, who's one of the most prominent HIV experts at the NIH and the National Cancer Institute. Now, this is data that he extracted from the New England Journal of Medicine. And the green line, it shows you the death rate for HIV, like how quickly people are dying from AIDS infections. On the red line, that shows the relative number of patients who are being given these protease drugs. And as you can see, in mid-1995, when many patients started getting HIV protease drugs, the death rate from AIDS drastically dropped. So once they added these HIV protease drugs to the cocktail, then AIDS patients were able to live much longer, more productive lives. Now, on the next slide, I'll tell you about what these multi-drug resistant mutant superbugs are. Infectious diseases, like HIV, almost always eventually evolve resistance against the different drugs that are used to fight them. To make it more difficult, HIV copies itself in a sloppy manner, so many changes or mutations occur each and every day within each and every infected patient. And if these accidental changes happen to help the virus resist the effects of the drugs, or if the lock's guts change and that happens to help block one of the keys, then those changes give that version of the virus a selective advantage, and it helps it spread and outcompete the other strains. But since these multi-drug resistant mutant superbugs keep evolving and spreading throughout the world, we need to keep discovering new and better drugs to fight AIDS. On the next slide, I'll present some recent progress from the results of Fight AIDS at Home Experiment 28. Now, in Experiment 28, we discovered 2 different compounds that are actually proven to help fight HIV protease. And one of these compounds here, APES1, shown in red, it's the name I gave it because I'm, Alex Perryman's predicted eyesight binder number 1. And the 1 is because the ligand ID number happened to start with the 1. Anyway, in red, I'm showing you this one of these new promising compounds we discovered on Fight AIDS at Home Experiment 28. On the left side, those 2 images show you that it's predicted to bind well to different versions of protease that have these closed flaps. On the upper right corner, you can see that it's also predicted to bind well when the flaps have a semi-open conformation, and it can get some atoms into that I site. Now, being able to bind into that I site of the semi-open conformations is a new strategy that could help specifically fight some of the mechanisms that protease uses to evade the different drugs. But you can see this little red compound is predicted to bind well to multiple different types of protease that have different conformations, and is a promising new anchor that we can then develop off of and try to grow and find bigger, larger compounds that will be much more potent. So right now, this compound only inhibits HIV protease activity by 45% at 1 millimolar concentration, but we need to find things that are actually 1 million to 1 billion times more potent before it can approach the activity of the current drugs. On the next slide, I'll show you the second new compound we discovered in Fight AIDS at Home Experiment 28. This has also been shown to inhibit HIV protease activity, and these studies were done by Ying-Chuan Lin and Professor John Elder's lab at the Scripps Research Institute. So we did the biological assays that showed that at 1 millimolar, this compound causes 50% inhibition of protease activity. So it's a little more potent than the other one, but we'll still need to develop this into something that's 1 million to 1 billion times stronger before it can be close to an actual drug. But it does represent a promising new scaffold or anchor point that we can then grow off of to find things that can hopefully counteract some of these mechanisms that protease uses to escape the drugs. On the next slide, I'll show you how we've extended Experiment 28 in order to find larger, more potent compounds. So in green, I'm showing you multiple different compounds that were predicted to bind well to this multi-drug resistant V82F ID4V mutant of HIV protease. And I'm showing the different compounds superimposed on each other in green because I can't yet reveal the chemical identity of these compounds before we publish them. So I have to publish them before I can reveal the exact nature of the compounds. But here I'm showing you that we found multiple new compounds that are predicted to bind very well and pack well in that active site region of this multi-drug resistant mutant superbug. On the next slide, I'll show you some other compounds we fished out from this extension to Experiment 28, here also shown in green. On the previous slide, it was a closed conformation of a superbug. Here, it's a semi-open conformation of HIV2 protease, which is the clade or group of strains of HIV that happen to predominate in Africa. So here you can see we've also found multiple different compounds that are predicted to bind very well in that active site, but also can place many atoms into that I site of the semi-open flaps, and thus represent a promising approach to fighting some of these superbugs. On the next slide, I'll show you or explain the new target that we're also focusing on Fight AIDS at Home. So in addition to HIV protease, we're also doing experiments against HIV integrase. Now, HIV integrase is another viral enzyme, and it processes the viral DNA and then permanently attaches that or integrates it into the human genomic DNA to cause a permanent infection of that cell. The HIV integrase drug raltegravir has been shown to help treat AIDS patients who are not responding well to the previous anti-HIV cocktails, presumably because those patients had multi-drug resistant mutants within them. Although raltegravir was approved in 2007, just a few years ago, there are already HIV patients who have drug-resistant mutants of integrase that raltegravir does not disable. On the next slide, I'm going to show you a very promising line of research that was done by Professor John Dedman's lab and published in Antiviral Chemistry and Chemotherapy this year. Now, this is showing you a new allosteric site and a new allosteric fragment that's been discovered against HIV integrase. Here I'm showing you that we can reproduce the binding mode of this known allosteric inhibitor. So I had to tweak the parameters a little bit in the docking calculation to allow us to reproduce the binding mode of this fragment, since fragments are often a little harder to dock than larger compounds. But we show here that the docked mode in green superimposes very well with the known experimentally determined binding mode showed in cyan. So after we've figured out how to reproduce the binding mode of this allosteric compound that binds in the region that's adjacent to the spot underneath the 140s loop. This 140s loop is known to be critical to activity. The active site's actually on the top there. But we showed that we could reproduce this binding mode, and now on the next slide I'll show you what we're doing for some current and future phytates at home experiments, where we're now docking to the region inside those blue and green boxes in order to find much larger, much more potent allosteric inhibitors to hopefully develop some new classes of drugs to fight the drug-resistant mutants of integrase. Now on the next slide, I'll show you what happens next. Basically, my contribution to this overall multi-lab program project is shown in that little red box, the computational coevolution. Then we collaborate with multiple other types of labs like the Torbit lab and the Elder lab to do the biological assays to show whether compounds actually work. The Stout lab in the bottom right corner, who does the crystallographic studies that can prove exactly where a compound binds and how it binds. And then we pass that data off onto the Sharpless, Finn, and Fulkin labs, or the medicinal chemistry people who can then try to optimize and develop these compounds into much more potent things that could actually be drugs. On the last slide, I need to thank the different people who've contributed to this project. Of course, special thanks to the World Community Grid volunteers like you for giving us over 145,000 CPU years of time. You can join us at worldcommunitygrid.org. Need to thank Art Olson, Stefano Forley, Garrett Morris, Sargis Delakian, Ruth Huey, David Goodsell, all the members of that program project team listed there. Of course, the members of IBM. And a special thanks again to the donors who give us your free computer time. You can join us at worldcommunitygrid.org. And we'll go to the last slide for the question and answer period. Thank you very much for your time, and please help us fight HIV.
Thank you, Alex, for a very interesting talk about the Fight AIDS at Home project. I hope those listening who have not already done so will also contribute their spare computing power by joining worldcommunitygrid.org. The second part of this lecture is the question-answer session about the Fight AIDS at Home project on World Community Grid.